Hiya, welcome to the video on the cell cycle. So, the cell cycle is basically the cycle that all cells go through, and it just summarizes what they're doing at different times. So, um, we draw it as a circle, and then the circle can be split up into different sections. Okay, now the biggest section here, so going around from here all the way around back to there, is what we call interphase. Now, interphase is actually made of three other sections, G1, S, and G2. So we could split this up into, that's rubbish, look, it's not very evenly spaced, is it? So we could split this up into three further sections. So we could split that into 1, 2, 3. So rather than saying all of that is interphase, we could say that that section's G1, that one's S, and that one's G2. Okay, so all of that is called interphase, and interphase includes G1, S, and G2. Then the next section is called mitosis. So that section there is called mitosis. And mitosis includes 4 stages, P, M, A, T, PMAT, prophase, metaphase, anaphase, telophase. And then the final section is called cytokinesis. And that's when the cytoplasm splits in half, so you end up with 2 new cells. And then the cell will go back into G1 again. Get rid of that end of the arrow because it's going this way round left. So cytokinesis to there, metaphase to there. Okay, then it goes back into G1, S, G2, back to mitosis, cytokinesis, G1, S, G2. All right, so the cells go round and round in this cycle. So let's go through what happens in each of these phases. So the first one is interphase. So interphase, as we've looked at, consists of 3 phases. First one, G1, is a growth phase. And what's going to happen is the cell is going to grow. Okay. The other thing that's going to happen is that the organelles replicate. So we're getting ready for the cell to divide. So if this one cell is going to divide into 2 cells, then we need to have enough organelles for 2 cells. So we need to replicate the organelles. The second part of interphase is S, which stands for DNA synthesis. So if we're synthesizing DNA, that means we're making more DNA. And what happens in this is the DNA replicates. So it goes from a chromosome that looks like this to a chromosome that looks like this. All right, so this chromosome here, we say contains 1 chromatid. This chromosome here, we say contains 2 chromatids. All right, so they're both chromosomes, that they're chromosomes at different parts of the cell cycle. So in DNA synthesis, the DNA has replicated, okay, and we have got twice the amount of DNA, but we've still got the same number of chromosomes. Okay, you've just gone from kind of these half-size chromosomes to these full-size chromosomes. You haven't actually increased the number of chromosomes. The final stage of interphase is G2, so another growth stage. Again, the cell can grow. And the other thing that happens in that stage is the cell checks the new DNA. So we've replicated the DNA and it just double checks that that new chromatid of that chromosome is right. Okay, so it checks the DNA. Now, I just want to talk about chromosomes a little bit more before we move on. So chromosomes are made of DNA. So if you unravel a chromosome, it just kind of looks like a long strand, okay? And that is called chromatin when it's all unraveled DNA, okay? Chromosomes are made of DNA. So if you were to zoom in on this chromatin, it would be the double helix that we know. Okay. And finally, chromosomes exist in two forms, which is what I've just been talking about. Usually we draw them as X shapes, and that's the time when they are made of two chromatids. So this one here is made of two chromatids, one on this side, one on this side, and they're identical, and they're joined in the middle by something called a centromere. Now the other form, so this is form number one, if you like. The other form that they can be in is this form, where we've still got the centromere in the middle, but this time we've only got one chromatid instead of two. Okay, so we've got this form where we've got two chromatids and a centromere. The centromere is just joining the chromatids together, which makes the X shape, which is the most famous shape. And we've got this type here, which is only one chromatid with the centromere. Okay, so that gives us a summary of the chromosomes. So just to recap, if you unravel a chromosome, you get this like wool, effectively, like string, which is called chromatid, which is when it's all unraveled. Chromosomes are made of DNA, which is this double helix that we've learnt about. And chromosomes have two forms. They've got this typical type, which is like an X, which is made of two chromatids with a centromere holding them. And then we've got this other form, which is just one chromatid held by the centromere. Okay. Now, in our cells, how many chromosomes have we got? So all our body cells have got 46. Why won't it move? Have got 46 chromosomes. Okay, so all body cells have got 46 chromosomes, which you can see here in pairs. Okay, so normal body cells have got 46 chromosomes. Oops, spell it wrong. Chromosomes. Okay, or you could say 23 homologous pairs. Okay, now this picture here shows those 23 pairs of chromosomes. So you've got two chromosomes that we call chromosome 1. All right, that's a homologous pair. Homologous means that those two chromosomes in the pair have the same genes in the same places. So we call that at the same loci. Okay, you've got two chromosome number 2s, 2 3s, 2 4s, and you can see they're arranged in size order getting smaller. So the way we number our chromosomes is just by number up to pair number 22, and then the 23rd pair is either XX for female and XY for a male. Okay, so that's your 23rd pair, and those ones are obviously the ones that determine whether it's male or female. So body cells have 46 chromosomes, 23 homologous pairs. Now, the other type of cells we got that aren't what we call body cells are what we call sex cells. Now, another word for sex cells is gametes, and they don't have pairs, they just have 23 chromosomes. So they would just have one chromosome number 1, one chromosome number 2, one chromosome 3. They wouldn't have homologous pairs at all. Okay, if we go back to our cell cycle, where was it? Just here. We've talked through the 3 phases of interphase. Next, we're going to look at the phases of mitosis, and then we'll look at cytokinesis. So, mitosis consists of prophase, metaphase, anaphase, and telophase. So I'm going to just split this into 4. So we'll start off with prophase, then we'll do metaphase, then we'll do anaphase, and then we'll do telophase. Now, the purpose of mitosis is to replicate the nucleus. The nucleus is the bit that's going to divide. Okay, so at the start of prophase, all the DNA is unravelled. So inside the nucleus, all the DNA is all unravelled as chromatin. Okay, so the DNA is unravelled, and we call that chromatin. Okay, now what happens in prophase is the nuclear envelope breaks down, so the chromosomes can move, the DNA can move anywhere in the cell, and the DNA condenses into the shape of these little X-shaped chromosomes. Okay, so in order to go from this to this, the nuclear envelope has broken down. Going to need a little bit more room. Let's move out this line and move it down a bit. So the nuclear envelope breaks down. That's the first thing. And the second thing is that the DNA condenses into chromosomes. Okay. Then we go on to metaphase. Now, the metaphase stage is when all these chromosomes move to the middle of the cell. Now, I always say M for middle, and they're going to move to the middle of the cell, which begins with M. Okay, so the chromosomes move to the middle. A better word for middle. Middle's good because it begins with M, like metaphase, but the fancy word for it is the equator of the cell. And the thing that moves them is the spindle fibre. Spindle fibres move them around the cell. So they put them onto the equator. Then we move on to anaphase. And in anaphase, those spindle fibres are going to contract and pull the chromosomes in half. So where they were, oops, they're drawn the wrong way, where they were an X shape, we're going to literally break them in half, and half the X will get pulled to that pole, and half the X shape will get pulled to that pole. And they're being pulled along by these spindle fibres here. I'll tell you a little bit more about spindle fibres in a minute. Okay, so these spindle fibres are going to contract. So we did have spindle fibres here, I just didn't draw them. So we had spindle fibres here attaching onto the centromeres, holding them or moving them to the middle. Okay, and then the spindle fibres have got shorter and started to pull them to the poles of the cell, so the edges of the cell. So the spindle fibres contract, which pulls the chromatids. So it's broken the chromosomes in half, and one chromatid is being pulled one way and one chromatid is being pulled the other way. So it pulls the chromatids to the poles. Okay. Then finally, in telophase, we reform the nuclear envelope, and instead of having one nucleus like we had at the start of prophase, we're going to have two nuclei. So these four chromatids here will be in this new nucleus, and the other four will be in this new nucleus. Okay, so two new nuclear envelopes are going to form. Okay, and the chromatids are now called chromosomes. They just get renamed. Right. So the whole point of mitosis is that it's nuclear division. The nucleus divides, and you can see at the start of the process we had one nucleus, and then by the end of the process we got two nuclei. So our final stage is cytokinesis. So in cytokinesis, we're starting off with that cell that we had at the end of prophase with our 4 chromosomes in each nucleus. And then what's going to happen is that the nuclear or the cell membrane, cell surface membrane, starts to pull in. Okay, and then it pulls in so much eventually that it totally divides into 2 cells. So it was the nucleus that divided in mitosis, and it's the cytoplasm that divides in cytokinesis. And what we've got at the end is 2 identical daughter cells. Okay. Now, let me just tell you a little bit about these spindle fibres and about how we label a cell. So, you know, when we think about the Earth, we can call it the North Pole, the South Pole, and the equator. When we talk about a cell, we usually draw it the other way around, and we call the edges the poles, and then a line through the middle we call the equator. Okay, so when you're talking about chromosomes lining up in the middle, you can say they're lining up on the equator. When you say they're being pulled to the ends, you can say they're being pulled to the poles. And then we've got these spindle fibres. Now, when we learned about organelles, we drew these things called centrioles, something like this. Okay, now what's happening here is the centrioles are moving to the poles of the cell. So there are centrioles. And then the centrioles make these spindle fibres. And it's these spindle fibres that reach out to the chromosomes and can move the chromosomes around the cell. So they're what we call the spindle fibres. And the spindle fibres are made of microtubules. So you might hear the word microtubules. So we can put under there made of microtubules. Oops, sorry, spelling. All right, and that's the end of that video.